Hello, welcome to my poster. My name is Félix Racine-Brassard. I'm a master's student in the lab of Pierre Saint-Jacques at University of Sherbrooke, and today I'll be talking about acquiring insight into mutational processes of cancer through the analysis of clustered mutations, and more specifically, mutational signatures associated with these clustered mutations. So the first question you might ask is, what exactly are mutational signatures? So before we get to mutational signatures, we need to generate what is called a mutational profile that is right here. And this profile shows basically an overview of the mutation found in a cell. So as you probably know, our cells are constantly exposed to damaging agents that can lead to the production of mutations in the genome, as we try to show here. And these mutations can actually be separated into 6 different types right here that are shown here. So those simply represent the conversions. So here C is converted to an A and so on. And so if you take, for example, one type of mutation, we can then separate each of them in 16 different categories. And these categories are called contexts right here. And what are they? So a context, as shown right here, is basically the mutation itself and the nucleotide that is directly before the mutation and directly after. So if we get these contexts, we get 16 different possibilities that we come and place in the appropriate one in the histogram. And if we do so for every single mutation, we'll obtain the mutational profile right here. Once we have multiple mutational profiles, we can look at the patterns that come up most often, and using non-negative matrix factorization, or NMF, we can extract mutational signatures like I show right here. So this process has been done using data from the Pan-Cancer Analysis of Whole Genomes project, where they used more than 24,000 tumors from patients to extract 16 mutational signatures that are now contained in the COSMIC database. These signatures are also associated with a cause. It can be exogenous, like UV radiation and tobacco smoking, for example, or endogenous, like deficiencies in certain DNA repair pathways, like I show right here. What's interesting with endogenous signatures is that they can be used as biomarkers to identify tumors and deficiencies in certain repair pathways. But in order to use mutational signatures as biomarkers, we 1. Need to be able to detect them reliably, and 2. To explain what mechanisms are involved in creating these signatures. So the ideal way of doing both of these things would be to isolate mutations produced by a single mechanism. So how can we do that? Well, here, for example, we have 2 signatures associated with the activity of ApoBec enzymes, which have been shown to produce hypermutated regions in cancer. With that said, we would expect that these signatures should be easier to detect by only looking at mutations really near one another, which brings me to our analysis. Here, we took profiles from the Pan-Cancer Analysis of Whole Genomes project and separated them by tissue type. In this case, we only show results from uterus tumors. With this heat map, we tried to find groups of tumors with similar profiles and see which signatures were present. Each lightly colored group in the heat map showed tumors with highly similar profiles. So here we have group 1, group 2, and group 3. In group 1, we have tumors with intense ApoBec activity, as shown by the 2 ApoBec signatures present in the profiles. In group 2, we have profiles with no easily distinguishable signatures. And in group 3, we have tumors with signatures associated with a defect in the MMR repair pathway. So next, as I mentioned a little bit earlier, we hypothesized that cluster mutation profiles would help us to understand mechanisms used by the cell. By doing this, we tried to isolate mutations produced by the use of specific mechanisms. So we would expect by doing this that we would have a group of cells with ApoBec activity, but maybe also new signatures associated with other mechanisms that produce mutations very close to one another. In this panel, we conducted the same analysis as above. The only difference is that we generated profiles by only using cluster mutations instead of all the mutations found in the genome, so mutations that would be circled in blue right here. We defined these cluster mutations as substitutions separated by a maximum of 100 base pairs. The first thing you'll notice is that we now only have 2 distinct groups of tumors instead of 3. Also, all the tumors found in group A that shows ApoBec activity were originally in group 1 or 2. This is what was expected for group 1 because we already knew there was significant ApoBec activity in these tumors, but what was surprising was that ApoBec signatures revealed themselves in the tumors that were previously in group 2, which means that we could potentially use clustered profiles to better detect these signatures. Also, group B is made up of tumors from group 3, which were associated with MMR deficiency. It also revealed a new signature that has never been described. We think that this signature could perhaps point us to a mechanism used by the cell in order to compensate for a deficiency in the MMR pathway. If that's true, the specific mechanism could become a therapeutic target for tumors with that signature. Our next steps will be to apply this method to other tissue types, then investigate what mechanisms would cause the new signatures that we find, and finally maybe correlate these new signatures with sensitivities to certain drugs by using large databases like the DETMap. Thank you for listening and have a great conference.